Good morning, everyone. Thank you for joining this third RePo webinar. I hope you can see my shared screen with the image of the announcement of the RePo webinar. Okay, I can see it also. So today we will have as speakers Carlos Loucera and Andrés Aldana. They will speak about two different Python packages for drug repurposing. So we are very happy that we could join them in the same webinar, and I hope you can learn and enjoy a lot of the information that these two great speakers have to share with us. So I will ask you to, during the presentations, to put your questions in the chat or in the Q&A, or if there is no questions during the presentations, we will open the micros also for have a discussion after each of the presentation. So I will start by introducing Carlos Loucera, the first speaker that we will have in the today's webinar. Dr. Carlos Loucera obtained a PhD in mathematics and computation at the University of Cantabria in 2017. He has worked as a data scientist at the National Institute of Statistics and the National State Agency of Meteorology, as well as a research scientist in several industrial projects at the University of Cantabria. Nowadays, he works as a postdoc researcher in interpretable machine learning for systems biology at the Computational Medicine platform of Andalusia, where he coordinates the data science group for the real-world evidence infrastructure. So his main interests are interpretable machine learning for knowledge discovery in systems biology, drug repurposing in rare diseases, signal transduction for single cell, causal and statistical evidence with clinical real-world data, and generative modeling for electronic health records. So we are very happy to have you, Carlos Loucera, in the RePo webinars from the RePo-SUDOE project. So please go ahead with your presentation, Carlos. Thank you.
Okay, thank you, Felipe, for letting me share our package in this platform for the RePo-SUDOE community. So I'm going to talk about the DREXML tool. Okay, it's open source software, easily installable through pip. It's available for everyone. Okay, but I'm going to center in the general strategy behind the DREXML packages, then in the specific part of the repurposing part, because there is a lot of, as it's common nowadays in bioinformatics, there is a lot of tools and data sets behind these things, okay? So it's a general picture and then only the specifics about repurposing. So I am part of the Fundación Progreso y Salud. I have to say it, okay? Because they are the ones that pay me, okay? And I'm also part of the Institute of Biomedicine and the Center for Rare Disease Studies in the Ciber Group, okay, across Spain. So I will start by using the COVID-19 as a context to explain the general methodology, because in our minds, this was like how things should work, okay? Like the life cycle of water, but with data, okay? So how we can reduce data? First things first. I am a mathematician, so I have been working with the biologists in and people from biomedicine in the last few years, but my biological knowledge is limited. Okay, but being said that, this methodology could not be possible without all the experiments that have generated the data that we use. For example, one of the key data sets for our study is the transcriptomic profiles available for more than 20,000 samples in the GTEx project. So without the GTEx project, this could not be possible. The same for UniProt, HPO, etc., etc., etc. So in the context of emerging disease, in this case, we are talking about COVID-19 2020, so no vaccines. We are in this part of the pandemic where there is little knowledge, but each day we are producing hundreds of papers, okay, of different degree of quality. So the idea here, you know what drug repurposing is. So we want to find new uses to approve drugs that can be used for other conditions, okay? Our proposal is disease-centric. So we take a disease and we try to find which drugs could potentially help in the outcomes of this disease, okay? So in the context of a pandemic, in an emerging disease, we need a quick response. So drug repurposing is especially suited for this kind of scenario, okay? And our idea is that, okay, we don't know how these drugs could be connected with the mechanism of actions in the context of the disease, in this case, COVID-19. But maybe we can choose our knowledge about the disease, distill it from the literature, then our systems biology approach to infer the signaling activity, okay, of ascertain, in this case, what we are going to call the disease map, that can be translated as which functions could be define what a disease is, okay? What functions are important for this disease. And the idea here is, okay, that we don't have data for fifth-hour models because maybe we are at the start of 2020, we don't have big repositories of public data about specific cohorts of COVID-19. Okay, but here is where the magic happens because this strategy was originally devised for rare diseases where we don't have enough data and we don't have and we have little to no knowledge about the disease. Okay, so our idea is that maybe we can use public transcriptomic profile health databases of healthy people like GTEx, and then we build our mechanistic interpretation of the disease map that is extracted from the literature. We compute activities of the functions in this healthy data set. We take from the databases, in this case, for example, was drug bank, about the informations of which of known drug targets, and then we can infer in healthy people how these functions that are important to the disease, we can infer how the drug targets are regulating these functions, and from there extract information, insights about the mechanism actions of these targets over the disease map. So, as I have said before, this is in our case like how we envision the data, okay, the full process. So this is a data-centric hypothesis driven and validation, okay? The idea, obviously, as a lot of people from the medical community tries to scare people is not to substitute clinical trials, is that in the absence of data or evidences, okay, we can make more informed clinical trials if we are gathering as much knowledge as we can, as much evidence based on the data as we can. So it all starts with building a map of the disease. Okay, in this case, in the COVID-19, it was a really big effort driven by a lot of different communities and groups across the world. Okay, led by Marek from Luxembourg, if I remember well. Okay, so the idea here is that they are building this knowledge graph, this knowledge big map that connects the different parts of what we know about the disease. From this, they build a very big disease map that can be incorporated into computational tools. From there, we build a simplified activity map, okay, that is this mapping this knowledge graph to known pathway databases, and from there extract only what they call activities. For us is keeping activations and inhibitions, okay, in the pathways databases. Then with this disease map, we are going to build the activity profiles using healthy tissue samples from GTEx in this case. Then we incorporate our DREXML methodology to infer which targets could be relevant to certain parts of the functional profiles, the functional landscape of the disease. And then before going to propose this clinical trial, to submit this information, okay, to the cycle, we can go to retrospective cohorts built, in this case, in the population health database from Andalusia that incorporates all the information that patients gathered across the health public system in Andalusia. Okay, and then we can check in this retrospective cohort if we can assess using causal evidence modeling if the drugs proposed are, from a data-centric point of view and using these causal mechanisms, if they are actually doing something with a certain endpoint that we can build in the retrospective cohort. Okay, so this is like the big picture. Then these things are incorporated into the literature. This builds packages, this builds datasets, this builds a lot of information that can be already integrated. And this, in our ambition of the future, this can be run in full cycle. Obviously, there are a lot of restrictions to do this because how these public health databases work for doing experimentation, okay? But this is our ideal world, okay? So what we can see here is that everything was built in 2020, but the results were disseminated because different processes of the bureaucratic machinery, okay, across 21, 22, okay, even 23. But, well, if you keep the overall picture, this is what we intended. And this can be run systematically, okay? So here is an overview, a brief overview of the COVID-19 disease map, okay? That it was, well, a big community effort, okay? From there is a simplified map that can be incorporated into computational tools, okay? And then from there, we perform a even more simplified version of this that is what can be mapped, in this case, to the care pathway ontology, okay? this is work being done by the people that do this in our laboratory, okay, the more biologically driven mechanistic modeling, okay, from Kinsa Ryan, okay, and there is also web tools that she builds in order to the community to perform certain experiments, okay. So the idea here is that we are mapping this to keep only which effectors to pathways are important. So we are not taking the whole pathways, only the minimal functional units that are mapped, okay? We call these minimal functional units circuits, okay? So in essence, what we are taking is for each one of the vectors in these pathways, so nodes with out degree 0, we keep all the routes that can go to all the paths that can go to this effector, okay? And these are a minimal functional unit that we call simplified as a circuit, okay? And the idea here is that we have an algorithm that has been explained for the last 10 or 15 years, okay, in the laboratory and in Valencia, where our IP, our PI team of workers, okay. So the idea here is that we have a signal transduction that takes transcriptomic profiles and just propagates the information through the receptors to the effector subpathways, so it gives a continuous score for each one of the circuits that can be translated to specific functions, okay? So the idea is to decompose the pathways and keep only the minimal functional units that are related to this simplified version of the disease map, okay? And this is what we call our disease map, our mechanistic map, okay? So in essence, if we check what we have for disease maps, the difference between the rare diseases from our point of view and the emerging diseases is how can we build the disease map. In the case of COVID-19, there was such a big effort that produces an incredible and detailed disease map. But in the case of rare diseases, this is no longer the case. So what we do is try to gather as much information as we can of which genes are related, okay, to this disease with human phenotype ontology distances, to associated genes in different databases, like DisGenet or FANET, etc. Then we map this to KEGG, keep only those circuits that appear, these minimal functional units that are mapped and that has these genes. So it's a selection from KEGG, and this is our disease map. Then we have the problem, okay, in rare diseases and in 2020 in KEGG, okay, our model is fed with transcriptomic data. We don't have enough transcriptomic data for cohorts of the different rare diseases. And in 2020, at the beginning, it was also the case for COVID-19. So the idea here is, as I have explained before, can we go to healthy samples of tissues and learn them a mapping between the gene targets of known drugs that has already been approved by other drugs for other diseases, and map them using our mechanistic insights, a machine learning tool that is going to map these targets in healthy samples to the activity of the map that are continuous values for these functions that are important for the disease. And to do this, we build is like multi-input, as we have all the profiles for the different genes across several thousands of samples. And then we also have this inferred activity in the signaling space for the disease map, okay, using Hypatia, our mechanistic modeling tool. So there we have, okay, several outputs and several inputs. As many outputs as we have effector subpathways that can be selected using the literature and mapped to care, okay? And we have as many targets as non-drug targets approved. We put several filters, we use drug bank, okay? The most important one is that it should have been approved, the drug, okay? For other conditions, okay? So then we use safely values, okay, to infer what genes, what targets are important for each one of the specific disease map activities, okay? And from there is a selection process and a scoring process using the safely values, okay? And we try to use the last recommendations from the literature in the case of this use of safely values. Okay, so we try to do this to test our model under permutations to also to try to test two things. On the one side, what is typically tested, and usually the only thing that is tested, that is performance. But if we are inferring knowledge from this, we also test how this knowledge generation process is also stable under permutations. Okay? So now I'm going to talk about the specific methodology. Okay? This is from the DREXML packages as the last version, okay, because we have been iterating through 2018 with different rare disease groups, then COVID-19, now schizophrenia and other rare diseases, okay? So at the end of the day, this is what I have been talking about. So the first thing is to build a disease map, okay? For us, for the model, a disease map is defined to the circuits available to Hypatia that are relevant to your disease. Okay, how do you build this? It's up to you. It's up to you. We provide several ways of doing it that are very easy and the things that can be easily extrapolated to any disease. But if you have more information about your disease, you can even, if you want, build these graphs because maybe you have more information about your diseases that what is appearing in the care pathways. If you can build these circuits, okay, these graphs that are directed, and you know what is activation and what is inhibition, you can also build your own disease map and the model is going to function. But we provide just a series of common tools for initial testing without any knowledge. So we gather as many evidences of genes related, we map them to care. From this is what we call the disease map, and then we build the whole process, okay? We are working in a platform that is going to be run in the new cluster of computation of the CyberRare, okay, in the rare disease groups of the fever groups across Spain, okay. That should be available for everyone in the world, okay. That has a graphical interface using the web, okay. We plan to release this at the end of this year, but it was also planned for the last year, so be reasonable probably next year, okay? I hope. So, and with this, we want to get out of the loop because until now we have provided the tool, okay, the software. It's easy to use if you are a bioinformatician, okay, but we have been working with the groups, and now we want to get out of the loop, so the groups can do this by themselves, and only if there is something very complicated or something that needs something new to the package, then we intervene. This is the idea, and this is how I think that software should work. Okay, so that we can get out of the loop, work on other things, trying to be innovative and being vanguard as others are using our tools that are robust to the users. Okay, this is our idea. Okay, we are behind the schedule as always. Okay, but we plan to bring it to the community next year. Okay, so this is extracted from the paper. Okay, that was published in Computational and Structured Journal, Computational Journal. Okay. So the idea here, as I have seen before, we have these samples we are working on here on GTEx, but if you have other transcriptomic profile database, you can use it for healthy samples. Or even there is popular, I mean, there are other groups that are trying to use this with healthy and uh, disease individuals, patients, okay, in cancer using their own data sets. We are still looking at how to do it because it seems similar, but with cancer, everything is a bit more complicated because there are, it's like everything is important, relevant, etc. So we are trying to we are focusing on what we know that works, okay? But there are groups that are trying to be adventurous with our tool, okay? So it's open to experiment because it's open source, okay? So the idea here is that, okay, now we are going to start in algorithmic details, but very easy from the top level, okay? So we have this on the left side is the transcriptomic profiles of known drug targets, okay? So genes per samples. And on the right side, we have these activity circuits, okay? So also continuous values for the same samples, okay? That are inferred using Hypatia, okay? So we are going to build a model that takes a certain sample, sets of samples, and is going to divide it into a validation part and a learning part, okay? We want to build this end to end. So it's not that we take the whole data set and we say which genes are important. No, it's that we want to test also that our selection procedure is stable, okay? So first, and also to be in accordance to the literature with safety values, we are going to have this background data set that is the learning where the model is fit. Okay, so we divide the training samples in learning and validation. The training is for the fit of the model. From there, we build scores using safely values in a multi-output scenario. Okay, so at the end of the day, we have for each known drug target and each circuit of the map, we have these scores and for each sample, okay? And this is done using the learning as a background dataset to integrate in the supply values. The supply values are computed through this validation, okay? Now from there, okay, this is the fitting and explain that I have talked, okay? Then we select the top caddies, the 90% of the caddies that explain the variance for each one of the circuits, okay? And with this, okay, we are inferring what it is important. So at the end of the day, we have a selection procedure, what target genes are important, what known drug targets are important for each one of the circuits, and globally just taking what is important for any circuit, okay? And with the safely scores, we have also a way of scoring them, of giving a value. So we are saying, okay, this gene to this function, it activates it, or so it upregulates the function or downregulates the action. So what we are putting is the known drug targets in context of the functions that are important for the disease, okay? And this is done in an end-to-end fashion, end-to-end learning, and we repeat this schema. Okay, we take the full GTEx dataset, we perform 100 splits, of 50% of the data for testing, 50% for training. And this model is run for each one of the subsets in the training phase. So this full scenario that takes a lot of computation is built for each one of the 100 splits with the validation, a refit to the full training data set, and then an scoring on the test set. Okay? So the idea here is that with this resampling technique, that is it is extracted from the selection stability from Sara Nogueira, okay, for machine learning selection, feature selection, okay, we can build a lot of to gain information not only about the performance of our model in a hard scenario, because we are dividing it with 50% of the data, and we are also not inferring the sub-values with the full data set of training, but with a part of this data set, okay? And we do this full fitting for the 100 times, okay? Like a bootstrapping procedure, okay? So it is computationally expensive, okay? But we have insights not only about the predictive performance, that if we are inferring knowledge about with the model, we have to check predictive performance, but this is not enough. We have to go further. This is stability, okay, about the knowledge that we are extracting. With this background data set and inferring the Shapley values in another part of the data set, we want to be more faithful to the data than to the model, okay? So it's not either extreme of the other. It's that if you go the usual way, you are very biased towards what the model is seeing. Okay, so the idea with this background integration is just to have a more faithful representation in the scale between what the model says and what the data says. Okay, and doing this by target and circuit, but extracted from a global model that is fit, okay, in a multi-output, full multi-output way, okay, is just because we want to go from global to local to function to sample, okay? So this is how we can split the data, okay? I mean, this, the 60% should be the same process, the same length, okay? This is a noun error, sorry. So how we can, we have a non-drug target selector population, no? So it's a population of which targets have been selected globally and for each one of the functions. So we can perform the complete Nogueira stability test, okay? That says, okay, how well are your model doing with respect to the selection of variables, in this case, non-drug targets? Are they stable under permutations of the data? So it's not a random process, okay? At the end of the day, it's from 0 to 1. The closer to 1, the more stable your algorithm is. So we want to check also the performance of the knowledge that is extracted from the model, okay? So we have this predictor also population. This is the classical performance that we are going to check across all the 100 iterations, the explained variance through all the circuits and the whole, the mean, okay, of the across all the samples, the circuits, and the whole map. Okay, so so we end with a bivariate graph. Okay, on the x-axis horizontal, we have the stability. The closer to 1, to the better. We have the stability for the whole map. That is like the average. That is one of these points, and the rest of the points are just the circuits. Okay. So we have an stability for each one of the circuits that says, for these circuits, your selection procedure is stable or not, okay? Well, it's on a scale, okay? And in the y-axis, in the vertical axis, what we have is the classical performance, okay? Since we have several splits and everything is done in an end-to-end learning scenario, we can build error bars for each one of the metrics, okay? So now a bit about the results, okay? In the case of COVID-19, okay, this was, well, the paper was very well received, and what the reviewer says that was important, okay, is that, okay, we predicted most of the drugs that were being tested, okay, so if you take the idea that we are using regular healthy samples from tissues, I think that is a good point for our model that is predicting drugs that were at least tested so suspicious for certain biomedical groups. Okay, but also we can divide our model, okay, and recombine the results in terms of hallmarks of the disease, in this case from the COVID-19. And the reviewer says, okay, you can see that this chloroquine, for example, that was used, okay, but you see that it is affecting almost every function. So at the same time, we have this global, but also this local way of viewing it. So you say, okay, probably it's going to say bye-bye to the disease because it's going to say bye-bye to the patient, okay? So the key idea is that we are profiling the targets of known drugs, okay, known targets of drugs in terms of the representative parts of the disease map in terms of biological functions. So this is important to help our discussions and to guide future interventions, okay? So the idea, okay, to work with retrospective cohorts, we put infrastructure inside the health system, so there is no data leak. So we build an infrastructure in order to be able to perform real-world evidence studies using the population health database of Andalusia. And well, this was the COVID was the first scenario to test this infrastructure, okay? So we have this retrospective cohort, okay? We are in 2020 with hospitalized patients with a confirmed SARS-CoV-2 infections, and we check mortality in the first 28 days or 30 days as it was done in the literature in 2020, okay? And we have a lot of co-variables, okay, extracted from the health records, okay, from the diseases, treatments, analytics, etc. So the age, the sex, etc. Okay, so we can build a proper causal inference. Okay, so the idea here is that we build this infrastructure, we check, and we show that there is a significant enrichment of the drugs predicted by our model, that with those that have a significant effect in two axes, okay, in the axis of death endpoint or lymphocyte count progression, okay, in the hospitalization, using, okay, inverse probability weighting, and well, this is not important, but I mean, using traditional ways of doing it, but to a big cohort, okay? So in the case of rare diseases, as I have said before, we are in the same scenario, okay? It's practically the same from our point of view. Here, it's not that you need it fast, because you are in an emerging disease that is affecting the whole population, but in the sense that we have few data, we have few knowledge about the disease, okay, and we need a cost-effective response. So the idea working with these rare disease groups were, okay, we cannot build a big cohort of this disease. So can we use what we have available and then work with the groups to make experimental uh, experimental designs to test the drugs that have been predicted, so informed by our model. And this is how we have been working with the groups, okay? And we have very positive results. Yeah, I mean, this obviously this is from our initial strategy that it was, okay, we are going to treat all the rare diseases in the world. We are going to treat all the rare diseases that are studied in Spain. Well, we have resources to a few a few rare diseases, and we are going to focus on those that we have known groups, okay, that wish to collaborate with us. Okay, in this case, we have okay, with Fanconi anemia, as I have said before. Well, this is the above is not the biggest one. Okay, so but here we have two proofs. Okay, here is only using DREXML, but this was the first the initial rare disease that we studied before the pandemic, and then it was also experimentally validated in cell lines by another group. Okay, here what we see is just on the right side we have the uh, the circuits that can be associated to the disease, what we call the disease map. Obviously, it contains the Fanconi pathways, but also more pathways that could be related and associated through the use of DGNet and other tools, okay, other databases like Orphanet, etc. Here we have the non-drug targets. These are the ones that could be selected, and we have how the target is performing in terms of the function, okay, of the effector subpathway, okay, because this is the known pathway and then 2 points and the effector gene, okay, to differentiate it in which part of the pathway we are. These are the minimal functional units, okay? So then we can laser focus on certain parts that are interesting, okay? And so in the validation paper, they say, okay, we need a target that doesn't uh, affect mainly affect that is its main effect is not parts of the Fanconi anemia pathway, but they can be with this cytotoxicity. Okay, so here our model says that this known target, okay, EGFR, could be potentially can be affecting these parts of the disease map. We check that we are not seeing what the clinicians say, that it doesn't have to touch the Fanconi pathway. And well, the idea is that we can reproduce what we have said before and what they say in this cell line driven study with our model. Okay, so in the case of retinitis pigmentosa, also published last year, we have we have mice-driven experimental test and also on the retina, okay, and frozen retina, and we build this DREXML pipeline. There are certain parts that are modified in the sense of the disease map, because when working with the group specialized in the regional group in from the CIBER that they work at Valencia, they said, okay, what we are seeing here in this part of KEF is not what we know about the disease. So there is a few changes, okay, here and there, but then the full methodology is going to work. So this is what I want to say is that, I mean, it's not something close, is that closer, okay, that with, in this case, working with us, you can modify things and then run everything as is, okay? What we want to do is that these things that you have in your mind, with this web tool that we want to release this year or next year, you can do it without our expertise. Okay, this is our goal and how software should work in our experience. Okay, so the results were very good and was published last year. Okay, so we are very happy, and now we are working in schizophrenia with a group also in Seville, a group in recessive dystrophic epidermolysis bullosa that I have to read. Okay, it's hard to me. Okay, that validations, experimental validations are ongoing. Okay, and also in Rasmussen syndrome. Okay, and we have also been trying to work towards improving the software and the full pipeline. Okay, because here what we provide is the model, but for building the data sets, if you don't want to use the ones used as a background by the model, you have to go to this repository to extract this script, to this other repository to extract this other script, and you have to download this from here, this from Zenodo, this from this Genet. So it's a bit complex, okay? So what we are doing now, okay, is to build everything in a Nextflow pipeline that is going to be released this year or the next year, okay? So you have the possibility to do it from scratch, okay? So it's going to be easy to modify. This is done by our pipeline expert at the laboratory, that is Daniel Lopez, okay? And we also want, well, to make it faster because, I mean, if you only have one disease, waiting a few hours is not a problem. But we also want to build a big a big framework to explore what the automatic process can do for all targetable rare diseases. Okay, so for this it's going to take a lot of time because it's running this several times for each one of the diseases. So we need to improve the times, how we visualize the drugs, because now it's very target and disease centric, and then you can combine the drugs, but it's not implemented in the packages. Okay, it's easy to do, okay, by hand, by a bioinformatician, but it's not implemented in a way that is easy to do for a not bioinformatician. Okay, we want to incorporate more information sources. For example, Danny and Kinsa are working with to try to integrate OpenTarget and make it more reliable to certain things. Okay, so distributed parallelization and interactive web. Okay, to perform all the analysis, and when you click, you submit to the cluster the computational, and you only have things to visualize and to stratify the results as you want. Okay, and this is going to be dropped for the next year. That is multimodal integration, okay, with other kinds of omics. This have this have to wait, okay. So this is the past and the future, okay, from our laboratory. So, for example, Marina is now in ETH in Zurich. So, I mean, but we expect, I mean, to have everything released by the end of the year or next year, okay? And we are at the Hospital Virgen de Rocío, University Hospital in the south of Andalusia in Seville. We are over 40 degrees. I cannot sleep well for the during the night. So I mean, bear with me. I'm a little tired, so I hope that I keep the times, but I don't see the. Okay, so this is it. Thank you to all. Okay, I'm open to questions.
Thank you, Carlos. Thank you very much for this very interesting presentation about this work package and the functionalities and the valencies and the disease where it can be applied, and also due to the future perspectives that you have for this software, I would say. So with this, I would like to open the discussion for the audience. If you have questions, please write them in the chat or Q&A, or please feel free to open your micro. Questions for Carlos for his presentation. I can start, of course. I can always kick off the discussion. So if there are no questions in the time that the people may think about, I will ask one. Carlos, you mentioned the circuit, so the mechanistic modeling that you have with this DREXML. So do you actually, and since I'm very interested in the molecular modeling of proteins and interactions of drugs with proteins at atomistic detail, so do you actually have information about the targets that we can target by the drugs to treat the disease?
Okay, so first, what we use here is static information from care. So we have the directed graphs, okay, that goes from receptors to effectors, and this is what we call a circuit, okay. Then the mechanistic models computes the transcriptomic profile with any kind of response. So it's not a machine learning model, okay. This is a mechanistic model that says, okay, we are going to follow iteratively the directions of the graph. It starts with a signal, and what we use for put in each node is with certain rules, okay, we put the gene expression of the samples. And then it is computed using a formula that basically, if it is an inhibition, is going to reduce the activity of the node, and if it is an activation, is going to augment it, and it mixes them together. And then we keep the result of the effector node, and this is the activity of the circuit. Then, with DREXML, what we link is which targets, which non-drug targets, could potentially affect each one of the specific circuits. Okay? So this is the link that we do using machine learning. Here, and we have the direction in the sense that we are augmenting the activity of the function, or reducing it. If you, for example, have a cohort, okay, and you can do differential gene expression, you can also do differential activity expression. So you have these circuits that are up or down in your map of the disease. And then you have these links with DREXML that says, okay, this target goes to this circuit and is up and down. Now, you take the drugs that are affecting this target. And so if they are inhibiting it, it's like reversing the symbol. And if the total result is reversing the activity map, then you are okay, because you are reversing the activities of the functional activities that are dysregulated in your cohort. This, for example, has been done in the well, it's very hard to produce to say to me, but the skin rare disease, okay? This recessive dystrophic epidermolysis bullosa, and this is also the case in the Rasmussen syndrome, that they have a cohort. So we know the directions of the circuits, we know the directions of the targets, we can put the information of the drug and say, okay, we want drugs that don't have upside effects on the disease map. So they are reverting it. Obviously, it's very hard to find a drug that exactly reverts the full activity of the circuits. Okay, but parts of it, and if they don't have anything that is on the opposite, that is what you don't want, well, but this is a drug that partially is helping your patients. This is, I mean, what we hypothesize, and then they try to build experimental validations about it. Okay,
thank you. Following your answer, can you also have insights about drug combinations?
Well, you can directly by the model as it is now. No, it is lying. Okay, because we have tried to incorporate up to 3 drug combinations, but the computational time, okay, and complexity explodes. This is mostly because of the time complexity of the sub values. Okay, so we are going to in the next iterations, when we build this web tool, we want to move towards using simple models that are easy to compute. Okay, but what you can do is that since you have the targets, all the non-drug targets, you can put your drugs, and for example, suppose in the best scenario, you have a drug that do what you want with certain targets in a part of your disease map, okay? And in the other part, the targets have not been selected, they have very few activity, no? And then you have another drug, and you check in the matrix that the other circuits, the other part of the map, is affected by this drug, and in the part that was affected by the drug, they don't have any meaningful activity. So you can you can build a theory that these 2 drugs combined can affect the whole map, for example. But up until today, this is only our theories, okay? Because no group has checked this combination of drugs. So it is our, I mean, it's our, well, hypothesis, okay, that you can do it.
Yes. Thank you, Carlos. There are questions from the audience to Carlos. If not, let's thank again to Carlos for his presentation. Thank you very much, Carlos, for the presentation within the RePo webinars of the RePo-SUDOE project.